Okay, hello everyone. Today I'll be presenting our discussion of a 2019 paper by Lee and college, tilted something along the lines of predicting gene expression using XGBoost. My name is Tsabita, and this is a joint work with Hammam and Fathi. So, what's the problem? Measuring gene activity or gene expression across the human genome is expensive and slow, but researchers discovered something useful. There are about 1,000 special genes called landmark genes that already captured the pattern of the entire genome. The question is, can we use the landmark genes to statistically predict the rest? That's exactly what this paper tries to do. Why does this matter? Gene expression data is used everywhere for diagnosing genetic disease, understanding how cells behave, and more. But getting full data is expensive. Previous methods like linear regression and KNN tried to do this prediction, but they had limits in accuracy. So the authors proposed using XGBoost, a tree-based machine learning method, to improve accuracy, speed, and interpretability.
So for data, they use two types of gene expression measurements. The first one is microarray data and RNA sequencing data, pulled from large public database like GEO, GTEx, and the 1000 Genomes Project. These are real large-scale datasets with samples from many different people. Next. The method they use is XGBoost, which builds many decision trees and combines with them. So why XGBoost? Because compared to deep learning, it's faster and requires less memory, prevents overfitting more easily, and important for scientists. It's also it is interpretable. We can actually see that the input variables mattered most in terms of structure. The inputs were 943 genes and the outputs were 9,520 target genes. They built a separate regression model for each of the target gene. Next.
So the big picture idea is simple. Measure only 943 landmark genes, fit them into a model, and get prediction for 9,520 other genes. It's like saying, instead of measuring everything, measure a small subset and predict the rest. Next. Before training, XGBoost needs some settings called hyperparameter to be configured. The authors use grid search to find the best combination. For example, for one gene called chat, they found that 350 trees was optimal. Adding more trees beyond that point didn't improve the model, so it's just risk overfitting, and they stopped there. Next. Now the result, they compared 6 models on the same test set. Performance was measured using mean absolute error, which lower is better. XGBoost achieved an overall error of 0.282, which beat all competitors. That include DGX, a deep neural network specifically designed for this exact test. XGBoost beat linear models about 25% and beat deep learning by about 12% without even needing a GPU. Next. They also went deeper and compared model gene by gene, not just on average. For each of the 9,520 target genes, they asked, does XGBoost or DGX predict it better? The result, XGBoost wins on 91.5% of all target genes. The remaining 8.5% where DGX wins tends to be genes that very complex expression pattern, where deep network have a natural advantage. Next. A good model should be generalized. It should not just work on the data it was trained on. To test this, the author trained on microarray data from GEO, then evaluate on RNA sequence data from GTX, a completely different technology with a different measurement scale. Even in this cross-platform test, XGBoost still achieved the lowest error at 0.439 compared to DGX at 0.453. Next. One bonus advantage of XGBoost over deep learning is interpretability. For each target gene, you can extract a feature importance score that tells you which landmark genes contribute most to the prediction. For example, for the gene chat, cartilage protein, the top 3 predictors were GATA3, BCMT1, and GNES. This kind of insight is biologically meaningful and could guide future research. Next.
So to summarize the study's strength include better accuracy than older method, good cross-platform reliability, and model interpretability. On the weakness side, using 943 input features raises overfitting risk, and training 9,520 separate models is computationally heavy. So one suggested alternative is a multitask model that predicts all genes simultaneously, which could be more efficient. Next.
Finally, if this research were continued, which suggests using larger and more diverse RNA-seq datasets, exploring transformer or other deep learning models as comparisons, and testing the model on external clinical dataset to validate real-world usefulness. Ultimately, the goal is reducing the cost of gene expression profiling in large-scale genomic studies. Thank you. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.